Welcome, folks. This is the first of what will be doubtless many online videos and slideshows and so on in the new COVID-19 world. First, hope you're all doing well. Thanks for bearing with us as we work through this alternate way to deliver class. This is really not my plan. I didn't want to be sitting here at the end of a computer screen. I really like meeting you guys in person. With luck, you'll get in touch in the synchronous class time Zoom sessions. Send me email. We can do Skype sessions, whatever's helpful to you. I really want to emphasize that this shouldn't be on you guys to figure out how to get instruction and how to get the classes done. It's on us. Not all of us are great and experienced in this stuff, but we will certainly do our very best. And I really hope you'll reach out and ask for whatever help or support I can give, whether that's with internet or with slides or with articles, really whatever I can do. On to the content. What you're seeing on your screen is the brain of a zebrafish larva. So zebrafish are actually a pretty common model in neuroscience. They are used for studies of genetic engineering, kind of like Drosophila. The larvae have the great advantage, they are transparent. And so what you're seeing here is calcium imaging. Each one of those specks of light, each one of those big flashes, reflects the firing of an individual neuron or a whole bunch of neurons in the zebrafish larva brain. You should recall from our early classes that as an action potential progresses, sodium rushes in, sodium rushes in, sodium rushes in. At the very end, calcium rushes in order to release neurotransmitters. And so getting a map of calcium activity and calcium flux in the brain can be a pretty good map of what's actually going on in terms of firing. This is just particularly cool because first of all, you can see it. And second, you can get the whole brain at once. The larva is actually immobilized in a tank with water flowing past, so it stays alive and healthy and allows the camera to focus on the whole brain at the same time. Neat stuff. Obviously, we've been looking at senses. A fairly recent, you can see this is March last year, discovery is that humans may, as well as vision and hearing and so on, also have the ability, which we know in several other animals, to sense magnetic fields, sense orientation of the Earth. So the experiment here puts the subjects into a large Faraday cage, and the experimenters could manipulate the orientation of the magnetic field. So this is a very large electrically charged box, basically. This magnetic field is not that big. And so they were able to change the magnetic field and make it move around in various ways. None of that was perceptible to the subjects. Subjects reported, I just kind of sat in a dark room for a while. But if you look at the EEG, their brains could detect when the magnetic field was shifting, which direction it was shifting to, and so on. You might be thinking, and you certainly should be thinking, what about a possible compound here? We know that TMS, transcranial magnetic stimulation, can induce electrical currents. Could that be happening here? Is that what could be explaining the changes in EEG? And actually, the experimenters thought about that, ruled it out, and showed that, for instance, the change in magnetic field from north to south is different from south to north and so on. So it's actually pretty well done. What you see here on the bottom left are nanoparticles. These are magnetite, and the nanocrystals of magnetite, which is a compound that responds to changes in magnetic field. These are actually found in the sockeye salmon eye. And so these crystals are now also being found in the human brain, probably an organ that allows us to sense magnetic fields. All right, last time. So we were talking about not just senses, but also attention. How do we select for and filter which parts of our sensory perception get into conscious level perception? So things come into all of our senses all the time. How do we select what we pay attention to, what we are actually aware of consciously? And we noted the fact that you only have a limited amount of attention. When you decide to pay attention to something, you process it more deeply, you certainly have a better chance of remembering it. Stuff that's happening and that your senses are noticing, but that you're not paying attention to, gets processed less, is less memorable, may well never enter your consciousness at all. You would actually deny this stuff occurred, even though it did. The cocktail party effect, the fact that you can hear your name from across the room, indicates that yes, you are in fact receiving all that information at some point in your brain, but it's not getting to your attention. That matches what we've learned about the neuroscience and the biology. So for instance, vision comes through the optic nerve, synapses in the lateral geniculate nucleus before it goes to optic and occipital lobe processing. So there's a subcortical level, a subconscious level of information that's available to the brain, even though the conscious level may not be paying attention. Certainly things that you don't pay attention to can change. This is what we saw in the video. So the colors of things can change. I gave you the example of the experiment where some workers with a large wooden door walk between an experimenter and a person they're having a conversation with, and that allows the experimenter to switch out for a whole different individual, the subject generally doesn't notice that the person they're having a conversation with has just changed, even if they're a fairly different person wearing different clothes and so on. An example that you might be able to demonstrate right now, think about what object was added to the slide 3 slides back that had no reason for being there. And if you don't know, go back 3 slides, pause the video, go check. There's a picture of a backpack, and that had no reason to be there other than to try and illustrate some of this point. When I've done this previously, 2/3 or so of the class don't notice that kind of thing. It's just not something that was relevant to them. And so even though it existed, it was right in front of you, your eyes certainly saw it, it didn't reach consciousness. This is just like the gorilla in the basketball pass counting experiment. In general, a model for senses, for motor, for the integration of the whole sensory motor system, simple information comes in at the sensory organ, gets processed up, and I think you should be able to see my cursor on the screen here, gets up and up and up through association cortex, which is the highest level of processing. And then that complex perception translates by the frontal lobe into a decision what to do about it, what action to take. And that action to take then goes down and down and down back into simple. So simple muscle instructions, twitch here. So you start at simple, you go up to very complex, you process deeply, you have a complex response planned, and that complex response then gets turned back into simple muscle actions. The path of that information is shown here. As we've talked about, the parietal lobe is commonly involved. And so you're integrating all this information, getting visual, auditory, sensory information, sending that information to some of these motor association areas, so motor cortex, with involvement of the frontal lobe. So we talked about this last time. The information comes in, gets processed, goes to the frontal lobe, where it gets, okay, what should we do? What should our response be here? And then the information goes back out to the motor cortex. The integration of that information is not just lateral, it's also, for the right parietal lobe, bilateral. The right parietal lobe takes that information from both halves of the brain and puts it together to form this universal, complete picture of the surroundings of your body. These are individuals with strokes in the right parietal lobe, and they all have this hemispheric neglect, this inability to pay attention correctly to half the world, with the overlapping deep purple area here being the area of commonality, the area all patients had damage in. And so if you damage that parietal cortex, here's the damage on the right, you get this contralateral neglect. There's also apraxia. If you can't do voluntary motions, that can sometimes be from a consequence of damage on the left. Maybe there's an integration there between planning and desired output that we haven't quite tracked down exactly. But the illustration of the right parietal lobe is pretty easy. If you're attending to your left visual field, then it's coming in contralaterally, it's going to the right brain. But if you're attending to the right visual field, note that both halves of the brain light up. Both halves of the brain are paying attention, and in particular, the right parietal lobe is integrating those two halves. So it's paying attention to the whole world here. Total model of information flow in the sensory system, information coming in, whether it's auditory or visual or somatosensory, all going together, frontal lobe for planning, output to the motor cortex. And remember, we can mess with this. We can alter your sense of who you are, how big your body is, where you are, all that kind of stuff, really quite easily. It's very plastic, whether it's an experiment with video cameras or the experience of playing a cello. Experienced cellists, that instrument becomes part of their body in many ways. And remember, of course, this is bilateral. So the corpus callosum connects those two parietal lobes and allows you to have a complete perception, a complete integration of where you are and what you should be doing. Underneath the cortex, as part of your motor output, the cerebellum, the basal ganglia are both involved with coordination and integration of different movements. They can sometimes serve as a backup. So even if you've got some damage to somatosensory cortex, you may still be able to take the appropriate motor actions. And all these actions get learned into complex patterns. So if you want to be a sprinter, you'll learn to pump your legs and move your arms and lean forward and so on in a very coordinated set of muscle actions. When you learn that, it gets stored through the basal ganglia, perhaps using the cerebellum. And again, that memory is no longer consciously guided. I gave you the example when we left class last of trying to walk upstairs by only consciously directing your feet. It's really hard. Most of your everyday motor actions are entirely subconscious. They're entirely things you've learned as a complex program, and then you just execute that program. We also started to think about taste and thirst in the context of how your brain has information again that you don't have access to. In this case, calories. So if you're drinking orangeade with or without sugar, with or without calories, what you see is that various areas of your brain respond differently. Your amygdala shuts off a little if you give it calories, and your amygdala is going to make you fearful and anxious. So shutting it down is actually a positive emotional state. When you give it a soda that doesn't contain any calories, your amygdala gets mad. It was expecting this sweet drink that you're getting to have some calories, and now it doesn't. That's an irritation. Your amygdala gets a little angry. Likewise, your striatum, where you're going to be releasing dopamine. You release a bunch of dopamine, a bunch of pleasure from the calories. You don't get it from the non-caloric drink. Even though consciously, these subjects didn't know which drink they were having. Various areas of their brain were very much aware of, this drink has calories, this drink doesn't have calories. Which of the two am I getting? Am I happy or sad? This will be the point in a regular class where I pause and ask for questions. Obviously I can't do that in this format, but I very much hope you'll either be able to come to the synchronous class session or send me an email, or as I've really said several times, try to reach out any way I can, try to reach out to Corey, get the help that you need to make sure that the loss of the live lecture is as minimally impacting as possible. I'll post the set of slides that Rachel and Jake gave me. The rest of you, this is still a task that's assigned. You should create a slide set. Ideally, if you have the ability, create a slide set like this with a narration so that you can give us the information and tell us about it, as well as just having a slide set. So do the best you can, send those to me, I'll put them online. The due dates haven't changed. So the one from March 12th, obviously class was canceled. I'll post that now to go with this slide set. Moving on, new information. So homeostasis, and in particular in this case, thirst. How do we control and give feedback to the amount of fluid that your body contains? Water is obviously a critical factor in terms of how well your brain functions, as well as how well the rest of your body functions. And so we need to very tightly regulate how much water you contain and how much water your brain contains. We do that by drinking, right? We take in more water when we are thirsty. So somewhere in the body, there's a detector. It says, hey, I've lost some water. Here's my behavioral response. I'll drink. And then you get a feedback mechanism. The thing about the feedback mechanism is it really has to be very responsive and act much more before water gets to where it's going. So when you start to drink, even at the level of the mouth, there are feedback mechanisms that start to tell you, hey, you can stop drinking now, some water's on the way. If you waited until the water actually got all the way through your stomach, into your blood, all the way to your brain, now you'd have too much water. You'd have drunk too much, and you'd have to oscillate back the other way. So you need a feedback system that responds even before the water actually gets there. And this is common for most kinds of homeostatic feedback. You also want to be not too sensitive. Think about a thermostat. If you had a thermostat that was regulating your house temperature at exactly 70.27565 degrees Fahrenheit, it would never be off. It would either be heating or cooling all the time. And as soon as it got a little too cold, the heat would come on. And then it would get a little too hot and the cooling would come on. You don't want that. You want a range of acceptable values within which nothing's happening. Everything's just homeostasis at normal levels. So here, you have a feedback system where water is coming in, and there are satiety mechanisms, right? Satiety is the opposite of hunger, the opposite of thirst. It says, I've had enough. And so this behavioral mechanism of drinking gets turned off once you get to a level of water that's okay. Water is really critical. As I've said several times, your brain is basically an enclosed bag of seawater with a bunch of cells in it. And so the environment is really important. It's also important around the body. So erythropoietin. Erythropoietin is a drug that allows you to increase the number of red blood cells, which means you have more oxygen carrying capacity in your blood. For an athlete, that's extremely helpful. It means you can work your muscles harder, get more fuel to them. And in cycling in particular for a while, EPO as it was called, was commonly used by most of the top athletes to get an advantage. Was it cheating? Yeah, absolutely it was cheating. It also had the downside that it makes your blood thicker. More red blood cells increase the density, increase the viscosity of your blood. And under conditions where you're very hot and sweating, so you're losing fluid, this can actually be really dangerous. So a couple of the articles on the screen here, these are cases of death in athletes who had erythropoietin, so they had very thick blood. And when they got too hot, their heart simply couldn't keep up. And so they keeled over with essentially heat stroke. Their heart gave out and said, look, I can't pump this overly thick blood. And they died. So fluid balance throughout the body, in the brain, in the blood, is critically important. You have specialized neurons whose job it is to respond to thirst. And these are called osmometric detectors. So they detect osmotic changes, changes in concentration of fluid around the neuron. And what you can see here is that as there's less water outside the neuron, water therefore flows down a concentration gradient out of the cell, making the cell smaller. That physical shrinkage causes the axon, causes the cell to fire more. And so this is a thirst response. The cell shrinks, that's interpreted by your brain as saying, hey, there's not enough fluid, go drink something, and that triggers a thirst response. Those cells are located right around the ventricle, which makes sense. You want them to be able to detect changes outside the brain very rapidly before the brain gets hit. And so here's a blow up of the area right around the third ventricle. The two areas circled in red, SFO and OVLT, the subfornical organ, and the organum vasculosum of the lamina terminalis. Do I care that you remember that? Nope. I simply want you to know that these two areas give me the acronyms. And the point is they're set up right on the edge of the ventricle here. We could talk about the signaling systems and so on. I really don't need that level of depth. It's the location. It's the location right on the edge of this fluid-containing balloon that contains CSF. When you see a response is very, very rapidly. So this is what I was telling you about the feedback mechanisms being more rapid than fluid can actually flow. So on the top is a brain, this is an fMRI, and here are some of the areas right around the ventricles there showing very, very high activity in a brain that has a person who's very, very thirsty. You give them a drink. 3 minutes later, those areas are entirely shut down. There's no possible way that fluid can have made it from the mouth to the stomach, into the blood, to the brain in that short a time. Nonetheless, thirst is shut off. So the thirst shuts off in response to water entering the mouth and water entering the stomach well before it gets to the brain to avoid over-drinking. Here's the basic summary slide for thirst. Thirst is really pretty simple. So you've got these two sensory areas, subfornical organ and the OVLT, the organum vasculosum. The osmometric thirst we've just talked about, sensing concentration, primarily comes into the OVLT. So these are the neurons that are responding to how concentrated is your blood, how concentrated is your ventricular fluid, do you need more water to get things back to the right concentration. The other thing that's detected is volumetric thirst. How much total blood do you have? So you have sensors, for instance, in the heart that sense, hey, how much blood is there total to pump around? Do I have enough? Oddly enough, on the battlefield, if you have a wound that is causing you to bleed out a lot, you may not perceive the pain yet because you're very stressed out, you've got a lot of endogenous opioids, for instance, blocking that pain reception, but you'll start to feel really thirsty. And that's because your heart is noticing, hey, there's not enough blood here, which it assumes means you need to drink. Your heart doesn't interpret and your brain doesn't interpret that as meaning, hey, you're bleeding out. But that signal of thirst is very sensitive. It says, hey, there's not enough total fluid. So volumetric thirst, how much total fluid? Osmometric thirst, how concentrated that fluid is. All that information comes in and is integrated at the medium preoptic. So this is the single point of integration for thirst that controls the drinking response. And again, that's right on the end of the ventricle, right on the edge of the brain. It's actually the very edge of the neural tube as the brain's developing. So it's able to sense the exterior circumstances of the brain very effectively. And so this medium preoptic is the place where drinking is instigated. That's what sends the message to the rest of the body. Okay, go take a drink. This is an important thing for you to do right now. Like I said, thirst is pretty simple. That's the entirety of thirst. So should you be able to reproduce that slide showing the integration of the different thirst areas? Yeah, and have a sense of how homeostasis works in general. Much more complex is hunger, ingestion of food. And this slide is partly a joke, but it also serves to illustrate the really important evolutionary point, which is that our bodies have developed and our brain has developed over a period of history where food was generally scarce. So if you saw someone who was markedly fat, it meant they were really rich. They could hang around all the time, they didn't need to work, they were probably a king or a king's daughter or some priest or some person of very high status and wealth. Food was scarce. We spent a lot of time looking for that food. Today, for many of us, and certainly in the westernized, fairly rich world, almost the opposite is true. Food, calories are plentiful. They're often cheap. You know, the number of calories you can get for $1 is some enormous amount if you just buy Twinkies. And so now we in fact envy folks who are able to restrain themselves and stay thin. The tables have turned. This is not healthy. Obesity is a chronic epidemic. It causes many, many health-related issues, type 2 diabetes, heart attack, all kinds of negative effects from obesity. And it's very widespread. So here are some data going up to 2010. Obviously, things haven't got better in 2020, but this was the 20 years up to there, from 94 through 2010 in the US. This is the prevalence of obesity on the top, diabetes on the bottom. And what you can see is the two diseases go hand in hand and go from an average of maybe 15% at most of the adult population to more than 30%. So more than 1/3 of the US adult population is currently classified as markedly obese, a BMI of greater than 30. In some places, particularly in the Deep South, there are areas in the Deep South where 50, 60, 70% of adults are clinically morbidly obese. That goes along with having almost half those adults in some places as having type 2 diabetes. You know, there's greater than 9%. That's a marked underestimate now. In many places, more than 1/3 of adults have type 2 diabetes. That is, obesity-associated inability to regulate their blood glucose. That has all kinds of consequences. You lose nerve function, you lose cognitive ability. It's really very, very bad. I do want to remind you that this is not universal, right? Not all countries have as much food and as much wealth as the US does. And I'm sorry that pause is partly me closing the door to avoid the cats making noise. One of the perils of working from home. But it allows me to leave this on the screen for a second. Yeah. Realize there are still today tens, hundreds of thousands of people, particularly kids, starving to death in a world that has plenty of food. I think it's important to step back occasionally and just think about the greater context of some of these things that we're learning about. You know, generally rich, well-educated subject populations, not always true. I mentioned obesity and diabetes. It's not just that that's the problem. Even in the States, which is obviously the world's richest country, look at that top graphic. 2/3 of teachers see kids who come to school hungry. In some states, on the bottom, 20%, sometimes more, of households don't have a secure food supply. So that starving to death is not just overseas. There are people, and again, especially children in the States, who have difficulty getting or do not get adequate food supply. That's in spite of the fact we throw out 40% of all the food we buy. This concludes the political part of the class, right? But that's nuts. Yeah, we can do better. Some parts of food intake are studied really through not just how we taste and directly perceive food, but also from how we're fooled. We've talked about attention. We haven't talked about the fact that we are susceptible to lies from information coming in. This is an experiment using an MRI scanner where subjects were fed wine. So in contrast to those kids who can't get enough to eat, this is an experiment where undergrads are paid to go drink wine sitting in a scanner and see how their brain responds. The point here, though, was the wine came in either a bottle with a very high price tag and a fancy French label, or a very cheap price tag, and I think it was a bad Chilean label. And subjects' perceptions were assessed and their brain responses were assessed. And what was found was, as you might guess, the quality of the wine was exactly the same in both cases, but people reported far more pleasure and their brains had far more dopamine release and so on from a wine with an expensive label and a fancy French title. So your senses, particularly in the case of how much you should be enjoying something, can totally be fooled. That's also true for things like how hungry are you, what are you seeing? All of this can be manipulated. How do we decide what to eat and what impact does that have? That obesity change is certainly due in fairly large part to a change in our diet over the past 20, 30, 100 years. And things have not got better. Things have got worse in terms of what we're eating. Here's one example of a substance in your diet you might have heard of.
Wow. You don't care what the kids eat, huh?
Excuse me?
That has high fructose corn syrup in it.
And?
You know what they say about it?
Like what? Yeah. It's that it's made from corn, it's natural, and like sugar, it's fine in moderation.
Love that top. Well, thanks.
Get the facts. You're in for a sweet surprise.
Okay, the Corn Refiners Council is not a neutral source. What they say is strictly speaking true. It is made from corn. It does have the same calories as sugar in some cases. And fine in moderation, what does that mean? Well, not clear. What is clear is that if you look at the change in our diets, again, these data are out of date, but the trend has continued. Here's 1970. Almost all the sweeteners being used were basically sucrose, table sugar. A little bit of glucose syrup, a little bit of just glucose, that's dextrose, same thing. Very little anything else. Over the next 30 years, what changed was the great increase in the use of high fructose corn syrup. So a change in the sugar from either 50% fructose, 50% glucose, or entirely glucose to be greatly enriched in fructose. The consequences of that have directly led, almost certainly, to the increase in obesity. So it's an increase in high fructose corn syrup. You can see that graph here. Things have leveled out after about 1990. Sugar use itself has gone down, high fructose corn syrup has gone up. And the reason for that is that it's cheap. So fructose is cheaper than glucose in terms of how much sweetness you get for a given dollar. And so food manufacturers have taken advantage of this fact and put high fructose corn syrup into almost everything. Ketchup, mayonnaise in some cases, many of your breads, lots of breakfast cereals, all kinds of stuff now contain high fructose corn syrup. This is table sugar, this is sucrose. And what you can see is that it has two different sugar parts. It's a disaccharide. This 6-carbon sugar on the left is glucose. This 5-carbon sugar on the right is fructose. And so it's 50-50. I'll remind you now, I'll come back to this in a little bit, your brain only uses glucose. So your brain really cares about this particular and very specific sugar. Equally, it really doesn't care and doesn't notice fructose. And that becomes really important. Glucose is what causes insulin release. So if you give someone glucose and they have a normal response, blood glucose will go up, stay up for about an hour maybe, come back down because the body produces insulin. In a diabetic, that's the green curve on the top here, blood glucose goes up and stays up because they're not releasing appropriate insulin, not responding to insulin. Your brain notices glucose, it also notices insulin. Both those two things serve as what are called satiety factors. They help to make you stop feeling hungry. Here's the metabolic pathway for glucose versus fructose. So glucose comes in, gets metabolized. No, I don't care that you know the details of all these enzymes, that's not the point. But notice that glucose has a couple of steps here that fructose doesn't have. In particular, I'm going to draw your attention to this step here. Phosphofructokinase, commonly called PFK, is one of the key enzymes regulating glucose metabolism. And the key thing to notice here is over to the right. It's inhibited by ATP. ATP, adenosine triphosphate, is the currency of fuel in the cell. It's the thing that actually gets broken down to produce energy for cells. And so how this works is that when there's lots of ATP around, glucose doesn't get metabolized. This is negative feedback. When your cell is in a high fuel state, you don't metabolize more glucose. This makes sense, right? You don't want to keep burning fuel you don't need. Notice that fructose doesn't have that step. You take fructose into your body, it's going to get metabolized. You're going to put into some kind of fuel state. And if you're not burning it off, which most of the time you aren't, what will happen is it gets stored as fat. So when you take fructose into your body, it gets broken down and used either as energy or, more likely, stored as fat. This slide shows you the same thing. And again, glucose comes in, regulated by ATP, so you use less of it when you're in a high energy state. If you don't need fuel, you don't break down that glucose. Fructose, not so much. And what happens is it gets stored very effectively as fat, much more effectively than glucose does. So taking fructose in is more likely to lead to obesity. Glucose, on the other hand, is pretty actively regulated. So insulin causes glucose to get stored out of your blood. That's what insulin does. It takes glucose out of the blood and puts it into stores as glycogen in your muscle cells and a little bit in your brain, from where it can be released when it's needed by this other enzyme called glucagon. The way insulin actually works is it causes, this is a muscle cell or a fat cell, insulin binds to a receptor on the cell surface, and it causes what are called GLUT4s. These little purple kind of blobs here are stored inside the cell. And when insulin binds, again, don't worry about details of this molecular machinery. This is actually a whole bunch of what my lab works on. So if you happen to care, I can give you a 45-hour lecture on insulin responsive mechanisms, but I'm guessing you don't care. When insulin binds, these glucose transporters get sent up to the surface of the cell so that glucose can come through them and go into the cell. So when you have a meal and it contains some sugar, your body senses that. It releases insulin. Insulin comes along and pulls that sugar into your muscle cells and your fat cells to control your blood sugar level. That's how you avoid diabetes. That's how you keep your blood sugar under constant control. I really want to pause and ask for questions. This is frustrating. I apologize. I'm sure it's equally frustrating for you. Please give me feedback, not only on questions, but am I going too fast? Am I going too slow? Do you hate my accent? Do you want to see me talk? I thought about showing you the video, which I can easily do, of me talking as I'm doing this. I don't know whether that's helpful or distracting or whatever. I really appreciate the feedback. The place in the brain where hunger and satiety are primarily regulated is the hypothalamus. So the hypothalamus is responsible for all kinds of basic functions. You know, are you anxious? Are you going to fight? Do you need to mate? Are you hungry? And so there are two nuclei, the one closer to the midline, the ventromedial, so it's slightly ventral and slightly medial. The ventromedial nucleus is the satiety center. So this is the opposite of hunger. That's where the brain is saying, okay, I've had enough to eat, I'm all good, I don't need to go out and eat more. There were some pretty wild experiments done in cats where this nucleus was lesioned, the VMN was taken out. If you take out the satiety center, what happened was the cats were never satiated. They were always hungry. And in fact, because the hypothalamus regulates aggression, they were also always aggressive. And so you ended up with these 50 and 60 pound, hyper fat, hyper aggressive house cats who were just snarling and eating the whole day. Really quite nasty. In contrast, the lateral hypothalamus is one of the brain's hunger places. And so if you lesion this, you will feel slightly less hunger. In fact, there are other places in the brain where we sense hunger because it's obviously an evolutionarily really important sense. We want to know when we need food. But there are these two opposing nuclei for hunger and for satiety. And within those brain regions, there are neurons that respond directly to the level of glucose. So GE here is glucose excited. This is a recording of spikes, of action potentials. Each one of these little black lines is one action potential. And it's taken from a brain slice in a dish where an electrode is being placed into a neuron from the ventromedial nucleus. So what you're seeing is this neuron is firing fairly continuously in a fairly high glucose bath. So 2.5 millimolar is about twice as much glucose as is normally sitting in the brain. Then when you change that glucose level to 0.1 millimolar, a short while later, the cell stops firing. You restore high glucose, a short while later, the cell starts firing again. So it's a glucose excited neuron. It's a neuron that is firing more when there is more glucose around. You also get GI neurons, glucose inhibited neurons. So these do the opposite. When you're in low glucose, now they start firing. And when you restore high glucose, they stop firing. So GE and GI neurons, these are cell types that directly allow the brain to sense glucose and to say, hey, I have plenty of glucose, I'm not hungry anymore, or I have very little glucose, I really need to be hungry. And it really is just glucose. The brain is very selfish. The brain only can use glucose, and so it doesn't care about other kinds of fuel. It doesn't care about fructose, doesn't care really about fats that much. It really primarily cares about how much glucose you have, and so whether the brain can get enough fuel. So if you want to stop yourself feeling hungry, a very effective thing to do is take a glucose tablet, because your brain will say, huh, lots of glucose, great, thanks. Table sugar, which is half and half. Half of it is glucose, And this is the most common form of sugar you might find in various plants, sugarcane, beets, and so on. Our brain is used to getting this kind of sugar. So it's used to having 50% of the calories in the form that it can sense and respond to. The other half just go somewhere else. And the somewhere else is actually pretty bad. So what does fructose do? It goes through different glucose transporters. In particular, it goes through GLUT5. So GLUT5 is a kind of glucose transporter that isn't found in the brain. It's only found in the gut. And so when you take fructose into your body, when you eat it, not only is your brain not able to sense it and say, hey, I should be less hungry now. In fact, your brain doesn't even notice it because it doesn't get to the brain. All it does is go through your stomach and get stored as fat. It's very effectively stored in fat. It also doesn't trigger insulin. And so the second signal that your brain uses to say, hey, maybe I've had some food recently, I shouldn't be hungry, that doesn't happen. As a consequence of all of this, it'll be no surprise that consuming fructose-sweetened beverages really leads to obesity, really causes increased fat storage and so on. In general, if you're going to have something to drink that's sweetened, stick with glucose. Diet soda, we saw, doesn't work. Fructose certainly doesn't work. Stick with just basic table sugar. Gatorade is pretty good. Gatorade actually contains directly glucose, as opposed to Powerade. Powerade is fructose. So no one really should ever drink Powerade. It's just not a good thing. This slide is purely in there as a vocab slide. There are three terms that are very similar and sometimes cause confusion. So glucose is the sugar. Glycogen on top is the storage form of the sugar. It's basically a whole bunch of glucose is stuck together, and it's in your muscle cells and in some brain cells, in your astrocytes. And then glucagon is the enzyme and the hormone that opposes insulin. So insulin gets glucose into cells and stored. Glucagon makes it come out of the cells, particularly in the liver, to back into your blood to give you some more blood glucose. How exactly do we sense glucose? And I'm actually going to go into a little bit of molecular detail because I think it's really cool and because we know all the details. And a big part of the answer is this glucokinase, okay? So glucokinase is an enzyme. It's the first step in glucose metabolism. And most of your cells don't have it. Most of your cells have hexokinase, which does the same thing, but is basically not sensitive to glucose in the range that you have in your brain. So hexokinase is always working flat out. Glucokinase, in contrast, actually changes and produces more ATP when glucose goes up and down at the levels your brain actually sees. And so if you have a cell with glucokinase in it, which is just these specialized neurons, these GE and GI neurons, they will actually respond to changes in the level of glucose by making more or less ATP. And then, as you can see, there's a special channel in these cells. It's an ATP-sensitive potassium channel. So this is a channel on the surface of the cell that opens and closes depending on how much ATP there is. You probably should be able to work out from our discussion of potassium channels and polarization and so on, that ATP closes this channel. So when you have more ATP, it closes the potassium channel. Potassium can't leak out. So there's more potassium on the inside. So the cell is a little bit more positive and therefore depolarized a little bit. So when you have more glucose around, there's more ATP, that potassium channel closes, the cell is more likely to fire. And so this GE neuron, this glucose-excited neuron, is directly linked to glucose levels causing firing because of this ATP-sensitive potassium channel. Now, it used to be, this is 2002, that we didn't know about GI neurons. That's no longer true. In fact, I'll tell you about that in a second. But here's a summary slide, right? Glucokinase, this enzyme that is not hexokinase, changes how much glucose is used and how much ATP is made in this range of concentrations. So there's about 1 to 2 millimolar of glucose surrounding your brain cells. That level is a level where glucokinase changes how much ATP it makes. That ATP then is produced by more glucose. You get more ATP, closes that potassium channel, makes the cell a little bit more excitable by keeping some of those positive changes on the inside, and therefore the cell fires. And that's the direct link between how much glucose you have in your fluid surrounding the brain, in particular in your blood, and how much these cells are going to fire to tell your brain, hey, am I hungry? Am I not hungry? This GE cell is going to fire more, and it's going to say, nope, I'm not hungry. We've got plenty of glucose. Everything's good. I mentioned that we know the GI mechanism. It turns out these same mechanisms are also used in the pancreas. So this is a picture from a pancreatic cell, a pancreatic beta cell that releases insulin. What you can see is that all of this mechanism stuff, glucose coming in, glucokinase metabolizing it, ATP going up, potassium channel closing, all of this stuff is exactly the same in the pancreas as is in the brain. It's just the consequence in the pancreas is that you release insulin, which makes sense, right? When you have more glucose coming in, you want more insulin to be released to cope with that insulin. But the mechanism is exactly the same in a hypothalamic neuron and the pancreatic beta cell. The body is very good at stealing a mechanism that works in one place and using it somewhere else. Here's the GI mechanism. So this is the slide showing that here's the ATP, again, being produced. And in fact, this time it's activating a chloride channel. So this is effectively an ATP-sensitive chloride channel. When there's more ATP, chloride can leave the cell becomes more negative, and so the cell is inhibited. This is a glucose-inhibited cell. You'll notice it's labeled CFTR. That's actually the cystic fibrosis transporter. So this is the channel that's encoded for by the gene that goes wrong in cystic fibrosis. The earliest tests for cystic fibrosis, basically the doctor would lick your sweat to see whether you were salty enough, because often you couldn't get chloride into your mucus or into your sweat. And that's why you died, because all this chloride was left behind. You didn't have appropriately regulated secretion of mucus. It was too thick or too thin, and it clogged up your lungs. So this transporter, this chloride transporter, is actually what goes wrong in cystic fibrosis. And it's the channel that exists on GI neurons in the hypothalamus, so that when there's more ATP, this chloride channel closes, more chloride is kept in the cell hyperpolarizes. I think I may have said open a moment ago. If that's so, that's an error. Just like the GE cells, where the ATP closes the potassium channel, in the GI cells, the ATP closes the chloride channel, keeps chloride in the cell, makes it more negative, and therefore doesn't fire. It's glucose inhibited. I mentioned a while back that insulin and glucagon are the two major hormones involved in glucose regulation. And we've been talking about homeostasis in the case of thirst. There's also this homeostasis in the case of hunger and blood sugar. So you really want to maintain blood glucose around 100 mg per deciliter or so. Gives you enough fuel for your muscles, doesn't cause nerve damage by being too high. When it gets a little too high, your pancreas senses that, releases insulin. Insulin pushes glucose into the muscle and liver. You come back to normal. Conversely, if blood glucose gets too low, now your pancreas again senses it, but in this case it releases glucagon. And glucagon tells the liver, hey, release some glucose stores, bring blood glucose back up. So this homeostasis is the balance between insulin on the one hand and glucagon on the other hand. This slide shows you exactly the same thing. It's a feedback system. There are actually two sensors, insulin and glucagon. So when you have too much high blood sugar, you trigger insulin release to come back down. When you have too low blood sugar, you trigger glucagon release to come back up. And all this is regulated at the level of the pancreas for hormone release, and at the level of the liver for converting glucose to and from this storage, to and from this glycogen. All right, I've been talking for almost long enough. We'll have a pause in a second. I want to point out, though, that your brain, although it's pretty selfish and responds only to glucose, that's not true for the rest of the body. Places like the liver, for instance, the liver does detect other forms of fuel, and in particular, does detect fat, so that you get an input from the liver coming up to the brain that tells you not just about glucose level in your diet, but also about fat. So if you eat 3 pounds of cheese, your brain won't notice that directly, but there'll be various signals coming up. This overly complex slide, don't try to memorize it, just illustrates that sugar in the form of glucose and fat and protein, some of the amino acids, all influence these cells in the hypothalamus and all affect the ratio of ATP. So your brain does integrate these signals. And in fact, integrates a whole bunch more of them, things than that. Various different hormones, lots of influences all coming to affect these ventromedial hypothalamic neurons. Don't try to memorize this. What I want you to get from this is the overarching concept that although your brain responds primarily to glucose, you do get input from other fuel sources. And I'll show you that again in a second, probably two or three slides time. I've put in here a couple more vocab slides just to help you read the text and read papers. I'm not worried about details here. Here's that summary slide I mentioned about all the different influences coming into your brain. So we talked about glucose. What other signals do we have? We have both short-term and long-term signals. Long-term signals, adiposity. What's your overall fuel state? Do you overall have a large amount of fuel? Essentially, are you fat? Do you have a big energy reserve? If you are, that fat will constantly be releasing and modulating a couple of hormones, insulin and leptin, and that information will go into the brain via the bloodstream. Short term, satiety. Have you eaten recently? Are you currently hungry, or is your stomach currently full? Have you had food in the recent past? And so your liver senses some of that, but also there are signals in the GI tract in the stomach. Some of them are mechanical. Is there physically food stretching your stomach? If you drink 2 liters of water before you eat dinner, you will stop eating sooner. CCK, that was in the vocabulary slide a moment ago, cholecystokinin, responds directly to protein and fat. Have you had a high quality meal recently? And all this information gets taken up by the vagus nerve and sent into the brain via a slightly different pathway. All of this information then gets integrated. So you have catabolic pathways and anabolic pathways, building things up, anabolic, just like anabolic steroids build muscles. These are anabolic pathways. So if anabolic pathways are being activated, and that uses the neurotransmitter NPY, then you're in a state of storage. You're in a state where the brain's saying, nope, got plenty of fuel, don't need this stuff, you can go ahead and store it. The opposite would be a state of hunger, catabolic pathways. Nope, don't have much fuel, really need to burn some. And there's a neurotransmitter called POMC, pro-opiomelanocortin, that characterizes the catabolic path. So again, two opposing states. Are you in a state of hunger, where you really need more fuel? Are you in a state of satiety, where you can afford to rest and replenish and build up reserves? Same information, different way, right? So long-term signals, short-term signals, all of them coming in. And ARC here, it was on the previous slide as well. It's the arcuate nucleus. ARC here is the arcuate nucleus of the hypothalamus. So I've talked about the satiety center and the hunger center. The information from both of those gets synthesized and put together in the arcuate. The arcuate, A-R-C-U-A-T-E, I'll put on the slides, is the final integration point in the hypothalamus for control of feeding, control of satiety responses. What makes us overeat? If we are so effective at regulating hunger, and we certainly are, why are we not quite as effective always at not eating? One answer is that we don't just eat because we're hungry. And in fact, most of us are very, very, really genuinely hungry. Most of us eat for social factors, because our friends are eating, or out of habit, because it's lunchtime, or for pleasure, right? Food is not just a thing we eat for fuel. Food is also a thing that we eat because we enjoy it. And so here on the screen, what you can see is a measure of one reason why some people eat more and get fat is because they don't release the same amount of dopamine. They don't get chemically the same amount of pleasure from a food stimulus. So what you're seeing is binding for dopamine. So the amount of dopamine being released and the amount of dopamine binding capacity in the brain, either of normal, non-obese folks or obese folks. And what you see here is there's much less dopamine binding in the obese brain. What that means is that, say we give you a donut. The control brain pumps out a whole bunch of dopamine. Yeah, this tastes good, it's sweet, it's got a lot of calories. Wow, I don't need any more, I'm all good. The obese brain, on the other hand, says, oh, I mean, it's nice, but I'm not as excited. I don't have as much dopamine being released. I don't have as much dopamine binding. Maybe I need 2 or 3 donuts to get the same level of pleasure as my thin friend over here gets out of 1 donut. And so there really is a case where you eat to achieve a certain amount of pleasure. And so if that requires you to eat 2 or 3 times as much because your brain doesn't have as much dopamine binding, then you're going to gain weight. And certainly amongst very obese individuals, I mean, these are folks with extremely high BMI, there is a pretty decent relationship. The less dopamine you release, the more food you tend to eat, the heavier you tend to be, because you're eating for a certain amount of pleasure. All right, this has been 48 minutes, which obviously is less than a normal class, but I'm going to stop it there. We've reached a pretty decent stop point. And also, I really, as I said before, would appreciate feedback in terms of, is this too long? Can you take it in? Was I comprehensible? So for the first one, we'll go short rather than long. I really appreciate you paying attention, and I'll talk to you soon. Bye, guys.